Now, our last but not least technical talk for today comes from Professor David Gifford, who really has an amazing track record in computational biology. He's one of the pioneers in this area from MIT. So David, I see your slides are there, so I would let you start.
Thanks very much, Regina. I'm going to be talking today about a new paradigm for vaccine evaluation, design, and its application to SARS-CoV-2. And I think that we're all familiar with the virus. This is a transmission electron microscope picture of the virus, and you can see how it gets its name from the crown around the coronavirus. And these are the spike proteins we're going to be discussing later on. And the amazing affinity for this spike protein, for the ACE2 protein that's on the surface of lung cells, is what makes this thing so darn contagious. And we're going to be talking about how to provide immunity to this virus in contrast to how to deal with it once somebody gets it. And there are three primary innovations I'm going to discuss today. We have a new way to actually model peptide HLA immunogenicity based upon data from convalescent COVID-19 patients. We then apply that model to evaluating vaccine coverage based upon HLA haplotypes in the population, so we can estimate the population coverage of a vaccine and who is not being covered. And finally, using that objective function, we can do combinatorial optimization of peptide selection for vaccine design. And just as background, typically this kind of peptide selection has been done manually using Excel spreadsheets, so this brings a new dimension to how to actually do a vaccine design. And the primary results today I'm going to communicate to you are, first of all, that the current subunit vaccine designs that are being promoted, we really believe need augmentation to provide further durability and effectiveness in older populations. And secondarily, that will show that the design methods that we present are superior to 29 baseline methods. So let's have just a quick review of immunology so we understand the adaptive immune system, which protects us from viruses like SARS-CoV-2. There are two aspects to it. There's cellular immunity, and then there's antibody or humoral immunity. And what you can see in this diagram is the cellular immunity represented by the red and green T cells, and the humoral immunity represented down here by the antibody secreting cells. Now, most vaccines right now are being evaluated on their ability to produce neutralizing antibodies. That is, antibodies that bind to the viral particle and inhibit its entry into a cell and thus infection. We're going to be focused today on cellular immunity, and what's really interesting about this is that cellular immunity by itself can actually confer immunity to viral infection. Now, cellular immunity is mediated through the presentation of viral proteins on the surface of cells. There are two kinds of presentation we're concerned about. One is class 1 presentation, which occurs on dendritic cells and also on virally infected cells via little billboards called MHC class 1 HLA proteins. And one nuance of this is that we all typically have different alleles of these proteins which control the specificity or what bits of the virus we're going to be displaying and actually use to activate our immune system. A second aspect of the cellular immune system is the helper portion, the CD4 helper cells. They're activated by the display by dendritic cells on MHC class 2 proteins, also little billboards. And these can actually assist antibodies secreting cells through cytokine interactions. So this is a very important system, and we need to understand when we're designing a vaccine how the bits of the vaccine are going to be displayed on these platforms and how they can actually induce memory. That is, memory CD8 cells and CD4 cells are going to help us later on when we are potentially exposed to the virus. And I think that we might all say, well, gee, a vaccine's a solved problem. There are 200 candidates out there. What's going on here? And what is interesting is that it's now known that the neutralizing antibodies in convalescent COVID-19 patients really are not very durable. Between 13 and 40% of patients lose these neutralizing antibodies. We've already observed reinfection of SARS-CoV-2 in individuals with different strains, and we know that coronavirus reinfection is common. It's been known now that the cellular immune system by itself can independently confer immunity to SARS and most probably to SARS-CoV-2. And emerging vaccine clinical trial data shows that not all vaccinated individuals actually have their cellular immune system activated. And finally, all Project Warp Speed vaccines share a risk, which is they all use the spike protein, which is a common immunogen. And I think that you've heard about differences in vaccines, and that's primarily differences in delivery. So whether or not you deliver the immune via messenger RNA, or you deliver it via viruses, or you actually deliver the native protein, you're delivering the same team. And the question we have is, how can we improve this team to better win the battle against SARS-CoV-2? So we're going to approach principal metrics for vaccine evaluation in 4 steps. The first step is we're going to characterize what parts of a patient's immune system are activated when they're infected by SARS-CoV-2. So we take blood from infected patients that are now convalescent, and we're going to assay them and ask which particular bits of the virus activate their T cells, indicating that those T cells have seen and been activated by that viral component before. So we split the blood sample from a patient into 11 different pools, and in each pool we're going to allocate a number of peptides. Each peptide is allocated to 6 unique pools. And thus, in this particular assay, we're going to assay 545 different bits of the virus or peptides to see which ones are immunogenic. We select the activated T cells in each pool, and then we sequence the T cell receptors. And any T cell receptor that pops up in 6 pools, we can map back to the activating peptide. This is part of the multiplexed identification of T cell receptor antigens assay, and we're going to use this data to build a model of what peptides are immunogenic in COVID patients. So when we take these data, we have observed in the assays 111 patients for class 1 epitopes and 8 patients for class 2 epitopes. And within this patient population, we observe a certain set of HLA alleles, as you recall, have a particular preference for which bits of the virus they display. And we also observe a certain number of peptides for which we've designed the assay. And when we build a machine learning methodology for predicting peptide HLA immunogenicity, we can get AUCs between 0.7 and 0.9, as you can see down here. So we're going to use this model then of peptide HLA immunogenicity, and we're going to use that to score how well a given set of HLA alleles is going to respond to the viral infection. This model then can be visualized in the following way. Here are some samples of the peptides and the class 1 alleles, and so the matrix essentially here for class 1 is 2,220 different peptides, and here are all the different HLA alleles, and a 1 bit here indicates that that particular allele is going to present that peptide and be immunogenic. So we can take this model then, and we can use it to characterize how a population responds to a particular vaccine. Now, the HLA genes are co-located together, and thus they're inherited together. And so you get 1 set of HLA genes from your mom and 1 set from your dad. And thus we're not going to use the marginal probabilities of inheriting a particular gene, but rather what's called a haplotype, or how often we inherit all these different alleles together. So you can see in the population that self-reports as white, this particular combination of HLA alleles is in 0.42% of the population, and for a population that self-reports as black, they're in 0.017% of the population. So there are significant differences in different populations as to the distribution of these HLA alleles. Now, since you got 1 haplotype for your mom and 1 haplotype for your dad, there are about 4.5 million possible diplotypes in our data for class 1 and about 2.9 million diplotypes possible in our data for class 2. And what we can do with a diplotype is we can examine how many peptides are displayed by that particular diplotype. Here we have a particular diplotype of different HLA alleles, A, B, and C, that are the classical class 1 alleles, and we can count the number of peptide hits that actually hit this particular set of alleles. This diplotype frequency is small, it's 0.004% of the population, but it produces a total of 6 peptide HLA hits that are then observed by the immune system. So we can then ask, what is the typical coverage of a vaccine by looking at how effectively it's covering a particular set of individuals. We can look at the count of the number of peptides in a given diplotype. So for all the different alleles in the diplotype, we can look at the number of peptides each of them displays, and we can sum them up. And then we can ask, for all of the diplotypes that produce exactly K peptides displayed, what is the probability of that by summing up the diplotype frequencies that have that property? And then when we wish to actually ask for a given number of alleles hits or greater, we can sum from n equal to k upwards. So that provides us with a principled metric for how well the cellular immune system is observing a particular vaccine. And so by point of reference, we estimate that a SARS-CoV-2 infection will produce on average 338 class 1 hits in your immune system and 3,707 class 2 hits. So the footprint then of a vaccine needs to be sufficient to be able to activate your immune system. And here are 29 different peptide vaccines that we analyzed as baselines. We can look here at this second panel, which asks for the population coverage. Here's 100% coverage and here's no coverage. And this is the number of peptides in each one of the designed vaccines we evaluated and their relative population coverage. You can see that these vaccines struggle to actually provide adequate population coverage at this particular number of peptide HLA hits. And this random is actually a random selection of peptides that we think are immunogenic, which actually does quite well here for class 2. So a question is, how can we improve this? Another question is, if we consider subunit vaccine designs that are being used in the warp speed candidates, and we count the number of hits they're producing against the immune system, what can we observe about their population coverage and what fraction of the population is not being covered by them? And so if we look, for example, at n greater than 10 hits for the spike subunit, which is used by all Operation Warp Speed vaccines, you can see that roughly speaking, about 3.7% of the US of the population is uncovered for class 1 and about 11% for class 2. The other thing you notice is that as the subunit bits of the virus that are used in vaccines get smaller and smaller, the corresponding amount of uncovered population increases fairly dramatically. So we would like to try and fix this. We'd like to actually provide better population coverage for the spike subunit. So how can we do this? How can we do vaccine design and select an optimal set of peptides to provide good coverage at a given number of peptide HLA hits? Well, we've designed a system called OptiVax to do so, and we start with either a set of peptides that are known to be immunogenic given the MIRA data that we have, or the all viral peptides, which results in a large set of peptides for us to consider. We also filter the peptides by their mutation rate. And we looked at 4,690 geographically sampled SARS-CoV-2 genomes to try and eliminate peptides that we think are going to undergo drift. The hypothesis is that things that actually have very low or zero mutation rates actually are functional and are not going to drift very much. We then can take all of our candidate peptides and feed them into a combinatorial optimization algorithm and come up with class 1 and class 2 vaccines to activate those respective parts of the immune system, or we can use a joint approach to come up with a single set of peptides that will do both tasks. So how do we do this combinatorial optimization? Well, what's interesting is that this optimization problem is not submodular. What that means is that a vaccine of size 10 is not necessarily a subset of a vaccine of size 11. Every time you add a peptide, you have to recompute the entire vaccine. So a very naive approach is to consider, say, all of the immunogenic peptides in SARS-CoV-2, and if you want to take 20 of them, you have to consider all possible sets of 20, which is 2,222 is 20, and for each one you need to evaluate it against all the possible diplotypes, and so you get a runtime that's order of 10 to the 55th, which is clearly not feasible. So we've come up with a new articulation of what we call a maximum n times coverage problem, and we have a beam search approach to this that gets it down to order 10 to the 12th, and thus when we do a typical vaccine design, it takes about 224 CPU hours to actually compute a single vaccine. So how well does this work? Well, recall previously we had these 29 baseline peptide vaccines that have been published for SARS-CoV-2, and for a given number of peptides budget for a vaccine, we can ask our OptiVax system to compute a vaccine design and then evaluate it. And what you see is that the blue line here is the frontier of our design, and essentially combinatorial optimization always wins. That basically you can always make a much better vaccine than has been designed before. The other thing that we have done is that we've started with a given subunit like spike, and then we've asked what can we add to that vaccine to actually make it better. And we add two different kinds of peptides. We can consider all possible peptides from the virus, or we can consider only peptides that are proven to be immunogenic and thus are more or less guaranteed to have a response in an individual. The dotted line here shows the peptides that are guaranteed to be immunogenic, and what you can see is that by adding just a few peptides to the spike protein, we can actually greatly improve the population coverage of that particular vaccine. And furthermore, we can improve the expected number of hits, which we think is going to be better to provide a bigger footprint on the immune system, to provide a potentially more durable vaccine and a more effective vaccine, especially in older individuals. So, experimental validation plans. We're working with a number of investigators here at Harvard and MIT to actually test variants of these vaccines. We're also using our model of how well viral peptides are displayed on different HLA backgrounds to use this to evaluate risks of patients based upon their HLA types. So I'd like to thank the people in my group that did this work, Guy Liu, Brandon Carter, and Sid Jane, and our collaborators Trenton Bricklin, and our collaborators from the NIH, Mary Carrington and Matthias Viard. One aspect of this work has already been published. There are two other aspects that are under final stages of peer review right now. And I'd just like to remind you that this is the first time that really a machine learning model of peptide HLA immunogenicity has been employed to actually evaluate vaccines in a comprehensive way given HLA haplotypes. And furthermore, we've used that objective function in a combinatorial optimization to do vaccine design to produce what we view are very good candidates. Our results are that we really believe that subunit vaccine designs should be augmented. We're quite concerned that the current spike vaccines are not going to be durable or effective in older individuals given the preliminary results from clinical trials. And we also have observed that these combinatorial methods are superior to all baseline peptide vaccine methods. Thank you very much. Appreciate the opportunity to present our work.
Thank you. Thank you very much, Dave. This is really very interesting talk, and we will have during the panel actually a number of people from the pharmaceutical industry who will talk about their efforts in vaccine development, and I hope that both Janssen and Moderna would have some direct comments on your talk. Do we have a brief question for David? So people are still typing. This will be our very last question. I'm trying to see. So far, I think I don't see the questions, but thank you very, very much, David. Really appreciate it. And good luck with your experiments. Thank you.
Okay, thank you.